Hello and welcome. Here's the audio recordings for Chapter 16, Host Microbe Interactions. A glimpse of history. So in this chapter we're going to be looking at how the microbes actually are designed to attack the host. The last two chapters we looked at was mainly looking at the immunological side of how the host is able to fight off these diseases. So we're going to take a little bit different approach here. And so in this history it's basically just talking about what people believed diseases were before we understood that they were caused by microbes. And so here we have the ancients basically thought that it was transmitted by contact, which was true for the most part. So at this point they actually took people that had leprosy and isolated them in their own colonies. Later on, as the plagues were taking over in Europe, people started believing that maybe they were contagious and it was best to stay isolated if you were sick. And then ultimately, it wasn't until 1546 that they started believing that things were caused by living agents, but then again, that was such a wild assumption back then, since there was no way to test it. And it wasn't until Leeuwenhoek came along with the microscope in the 17th century that they were able. Then ultimately Robert Koch offered proof of what is now considered the gold standard for understanding microbes causing disease, when he was able to show that actually the bacteria Bacillus anthracis was causing anthrax in people. So the first word that we see up here is ubiquitous. Ubiquitous just means they're found everywhere. So the idea that cars are ubiquitous on the streets is a way to use that term in a different way than just talking about microbes. So bacteria are ubiquitous, meaning that we have found them everywhere, from the Antarctica ices all the way through thermal vents in the bottom of the ocean. So microbes are just about everywhere. So we're breathing them in, we're ingesting them, we are picking them up on our skin all day long, whether you like it or not, they're there, but most of them are harmless, so they're not going to be able to cause any diseases. There's really just a very, very small percentage, somewhere in the 2 to 3% range, that actually cause disease in humans. And that's because of our innate immune system that's basically going to fight off a lot of these problems that we've seen in Chapter 14 with our innate system, where our skin cells are constantly sloughing off, as well as the stomach acid in our digestive system that's going to destroy a lot of these microbes before they even have a chance to establish disease. But the ones that are able to cause disease, a small portion, are what we call pathogens. So in reality, despite what we're told about bacteria, most microbes are actually harmless and many are beneficial. So we call this the normal microbiota. These are going to be ones that are supposed to be or typically reside on our body surfaces. But this is a very delicate balance, because the relationship between these microbes and our body is easy to switch into a different situation if the person finds themselves immunocompromised, whether it be from disease, cancer, alcoholism, drugs, or maybe just born with an autoimmune disease. Some of this balance can be thrown off in the favor of the pathogen, and we call these opportunistic pathogens, ones that typically don't cause disease, but in the right opportunity are able to. So this is what we consider to be the biggest issues when we're talking about normal microbiota. So we're going to see as we go through the rest of the series that a lot of these microbes that we consider normal microbiota actually can cause disease in humans given the right opportunities. So as we've seen in Chapter 14, the skin and the mucous membranes are barriers designed not only just to make our body not fall apart, but also they serve a purpose in protecting us against the microbes that we're breathing in every day or eating or whatnot. And so we can design these microbes, or we can basically categorize them in 3 different ways: mutualism, commensalism, and parasitism. And these words can be used not just in microbes versus hosts, but they can be used in a lot of different other avenues in life. So mutualism is when both parties benefit. Essentially, the microbe is getting something from us in terms of a place to stay, food, whatever it might be, and in turn they're supplying us with different molecules that they're able to produce that we're not able to get anywhere else, such as vitamin K and vitamin B. Most of your energy drinks are just loaded with vitamin B, and different derivatives of vitamin B. And mainly this is just something that microbes make as a byproduct and our body's able to use. Commensalism is when one party benefits but the other's unharmed. So this is going to be a lot about the normal microbiota that may be living on our skin. They're not really doing anything there except for taking up space, and that's the biggest thing. They're taking up space where no pathogen can actually sit down and cause problems. However, we're not gaining much else from them, but they are getting food and all the areas in terms of shelter to live on our skin. Now, the one that we worry about and all pathogens fall underneath is parasitism. This means that one organism benefits at the expense of another. So when it comes to the pathogens, they are actually chewing the host apart, more or less, and actually damaging organs, tissue, whatnot. And so there's really no benefit to the host whatsoever to have these on board. So we have the resident and transient microbiota, and this is what we talk about when we're talking about ones that are typically living on our skin, and those would be the resident. They're there for a long period of time, and transient may be there only temporarily. And this is actually becoming much bigger topic now that global travel has increased over the last couple decades. Mainly every area that humans inhabit, they're going to have different resident microbiota in those countries. And so if you go to travel somewhere, a lot of times people experience traveler's diarrhea, things like this, and it's really just your body readjusting to the microbiota that might be in that area versus where you normally are staying. So resident microbiota are going to be ones that are there for a long period of time. Transient is going to be pretty short period of time, typically when you're traveling. The Human Microbiome Project is a federally sponsored program that is looking into all the different microbes that survive on the skin and in the body, wherever they might be, just trying to figure out exactly what we have and what they're doing, if anything, and truly trying to understand everything that's involved. So the protective role we've already talked about a little bit, but mainly the normal microbiota provides significant protection over any of these pathogens. And as I said, some of these normal microbiota can in turn become pathogens if given the right opportunity. We'll see that as we go through this chapter and the next. So some of the things that the normal microbiota are doing that protect us is covering up the binding sites. In order to cause disease, one, we have to get the microbe into our body, and two, it has to attach. If it can't attach to any of our body, any of our cells, then essentially it's just going to be thrown through the system and out the other side. And so by covering up the binding sites, normal microbiota protects us against these pathogens. Another thing they're doing is they're consuming all the nutrients, so there's really nothing there for the pathogen to sit down. And it really comes down to numbers. When you think about bacteria and how fast they can grow, we're talking about that they can get into the billions and even trillions on our skin pretty quickly. And so each of those individual microbes needs something to eat, and so typically when you pull in a pathogen, you're only pulling in a few of them at a time. And they're not going to have the opportunity or chance to try to compete in that environment when they're just outnumbered by so much. Some bacteria, as you'll learn if you take cell biology or any of our biotechnology courses, some of these bacteria actually can produce their own what we call ancient immune system, I guess would be the right way. They're able to destroy a lot of other pathogens or other bacteria that come in contact with them with something called restriction enzymes, which again you don't need to know for microbiology, but it helps explain that bullet there. Now, so some of the things that we've learned is that the antibiotics that we give to people when they're sick with a known bacterial infection, they don't just target the pathogen. They're going to target a lot of these normal microbiota too, because they really are just all bacteria. And so by killing or suppressing these normal microbiota can actually open up our system to more pathogens showing up. So what we're going to see as we go through the rest of this course is that a lot of these microbes that cause disease actually are the secondary pathogens. They're not the ones that actually initially caused a person to be sick. And so this is something that causes a big problem in fatalities, especially in hospital settings or settings where healthcare may not be available, where the secondary infections end up being the cause of death in a lot of people. So For example, things that are a little less benign, lactobacillus is found in the vagina of females, and it's producing lactic acid, hence its name, basically dropping the pH in the vagina to a level that's almost too harsh for any other pathogen to grow, such as Candida albicans, which is a resident yeast, part of our normal microbiota. But if we take an antibiotic, it's going to take out the lactobacillus since it is a bacteria, while Candida albicans is a eukaryote, and so the antibiotic won't touch the yeast. Unfortunately, what that does then is it opens up a lot of area for the Candida albicans to start growing, where lactobacillus was typically halting it. And when that occurs, some females go through multiple yeast infections, or maybe only one or two in their lifetime, but essentially this is what we call vulvovaginal candiditis, and typically it's just caused by the lactobacillus being harmed in some sort of way. Some other ones, oral antibiotics. A lot of times oral is not the best route, because let's say you have a skin infection. What's the point in taking an oral antibiotic that has to go through the digestive system, be absorbed, and then finally find its way possibly back to the skin where the actual infection is? So sometimes oral antibiotics are the wrong choice, and they can actually block some of the normal microbiota that's found in our intestinal tract, and will allow some of the other normal microbiota, such as Clostridium difficile, to start overgrowing. And when that occurs, sometimes Clostridium difficile can actually start producing toxins, and when that happens, you end up having a serious disease. Stimulation of the adaptive immune response is talking about how if we don't actually stimulate the immune system on a regular basis, we can have problems with its ability to actually fight. It's sort of like the idea of going to the gym. You don't have to go to the gym, but if you do, you're keeping your body in shape and constantly stimulating the muscle tissue to stay as strong as it can. And so think of the immune system sort of like that. If we don't constantly have it exposed to microbes, it's not really going to know what to do when the time comes. And we've been able to prove this with mice, mainly mice that are raised in vivariums, which is a term we use for college or not, any place that has a research institute. The vivarium is where we keep the animals, and typically the vivariums are kept as sterile as possible. Of course, you can't sterilize an entire building, but they're kept as clean as possible to minimize any microbes, and there are actual rooms that are considered microbe-free based on some other steps that are taken in the protocol to ensure that. And so mice that are reared in those levels, they have very underdeveloped MALT. Remember, this is part of the adaptive immune system, essentially places where we find the macrophages and dendritic cells, just kind of checking things out along the digestive tract. And if these mice are never known to what a microbe looks like, when they actually do come in contact with microbes, they are not able to really do very much. And so this goes to a story that is something that I want you all to kind of think about, where parents essentially that raise their children with no exposure to the outside, maybe fear of germs, fear of them getting sick, whatever. We call this the hygiene hypothesis, which essentially you're doing more harm to the system than letting the child go around and quote-unquote eat dirt and help build that immune system up to a very strong level. And along the way, they're actually being exposed to a lot of the childhood diseases that are much easier to tolerate as a child than it is as an adult. So the fetus is kept by the placenta sterile up until the moment of delivery, and so a normal child that's born through vaginal delivery is exposed to a lot of microbes as it goes through the birth canal to essentially start coating the skin and whatnot with some of the normal microbiota, and this is going to help protect it right away from any pathogens once it's immediately brought into the world. And so other humans, everybody wants to kiss a baby, everybody wants to pick a baby up, everybody wants to hold a baby. Well, every person that touches the child is basically colonizing it with their own normal microbiota, and that's not a big deal if everybody's from the same area. This is going to basically coat it with the microbiota, coat the newborn with the microbiota that's found in those areas, and that's going to be typically a very good thing. So this is dynamic, as we already talked about. These changes can, in your normal microbiota, can change whether you travel or as you age. Sometimes your normal microbiota can change. Hormonal changes, especially in females as they go through menstruation every month, the hormones are changing, and so therefore this can cause different normal microbiota to either be killed or to increase their level of growth. It just depends on which pathogen or which bacteria we're talking about. And also your diet can be a huge difference. If you decide to switch to become a vegetarian, you're suddenly not going to have a lot of the microbiota that you might get from consuming meats. And when that occurs, well, then your normal microbiota is going to be changing. We've also got some indication here in the last few years that the normal microbiota also play a role in people that are obese or lean, and that we actually have tested and tried the idea of transferring some of the normal microbiota from somebody that's lean into somebody that's obese, and actually had them see that they start possibly changing their weight. And so we call these fecal transplants, and as gross as it sounds, it is exactly what it sounds like, but that's where we find a lot of normal microbiota, and so it makes a good starting point for attempting to try to force a person's normal microbiota to change. So when we talk about a pathogen actually getting into the system, into the host, and establishing disease, we call that colonization. And that's not going to occur until the microbe actually attaches and is able to successfully start going through binary fission. And so when that does occur, the term is colonization. So this is, instead of saying you're infected, usually we say, you've been colonized if we're talking about research. But when we're talking about infection, we're referring to strictly just a pathogen. And so a lot of infections are subclinical. This means that there's no symptoms, or the symptoms are so mild that you're not going to go see a doctor. And so these are going to be severely underreported because, well, of course, there is no reporting done on them. And so we have to kind of assume through different statistics how to come up with some of these numbers. Yet infectious diseases will always give us some sort of noticeable impairment, and we switch these around a lot, but we have to make sure that we keep these two terms in our head separated. This is symptoms and signs. Symptoms are subjective. This is going to be a patient coming in and telling you what's wrong, and you always got to remember that different people are going to experience things differently. So for instance, pain, what you may consider intolerable pain, somebody else may consider to be moderate. So in this case, sometimes the symptoms really just don't tell us a whole lot, but it is going to be something that the patient is experiencing and has driven them to come to the doctor's office. Signs are what we call objective evidence. This is something we can actually see, such as a rash, pus, or swelling. And this is somewhere we can actually start isolating where the infection might be coming from, or where we can start figuring out where to actually treat the patient. And so signs become much more useful to a clinical setting versus symptoms. The initial infection is what we call the primary infection. So for instance, somebody that might have HIV, that might be a primary infection, but as they start losing their immune system function, they're going to open up their body to more secondary infections, and these are going to be ones that come in afterwards. And these tend to be problematic because the immune system is not very good at actually fighting off two things at once. Just like, it'd be a very unfair fight if you had to fight two people versus yourself. And so in this case, sometimes the secondary infections are the ones that cause the biggest problem in people that are infected with something else that's chronic, such as HIV or hepatitis C, things like this. So the primary pathogen is the one that causes disease no matter what. If you're super healthy, live a great lifestyle, a primary pathogen is still going to cause disease in you. So this is going to be things such as Yersinia pestis that causes the plague, Plasmodium, which is malaria, measles, and flu, diphtheria, which we'll be covering in Chapter 21, tetanus and tuberculosis. All these will cause disease no matter how healthy the individual is. Now, an opportunistic pathogen is the ones that I've been referring to that are normally part of our microbiota, but given changes in our body in any sort of fashion can actually cause these to start forming a disease or forming a colony much larger, which then can ultimately start causing signs and symptoms in the patient. We refer to this as their virulence. So the virulence is, when we talk about a pathogen, we want to know how virulent is it. This is going to tell us how fast they're able to cause disease in a person, and not necessarily how fast, but what exact molecular mechanisms are involved for it to actually cause the disease. And so those mechanisms are what we call virulence factors, and so we'll be talking about a lot of virulence factors as we go through the rest of the chapters in this semester. Now, when we talk about the diseases, we usually talk about them being communicable or contagious. This just mainly means that they can spread easily from person to person, or sometimes animal to person, or person to animal. This is what we refer to as being contagious, or another term is communicable diseases. So the thing we need to know about that particular pathogen is how much of it do we actually have to bring into our body to actually establish an infection. And so this is known to researchers as the infectious dose. And the way that we do this is usually with mice models, and what we're looking for is what's called the ID50. And the ID50 is the infectious dose of 50% of the animals. And so when we have an ID50, what we're looking for is, say I have 10 mice that I inject with a particular pathogen, then we're looking for 50% of those mice, or 5 of them, to actually establish disease, where the other 5 are able to fight it off. So two related bacteria, Shigella and Salmonella, they're basically cousins. Their ID50s are completely different. For instance, Shigellosis, which is caused by Shigella, is one that you only need to ingest a few of the bacteria to actually cause disease. And one of the biggest reasons why this is that Shigella is able to survive the stomach acid, and so it's able to get right over to the intestine. So we'll be seeing this in Chapter 24 when we start going through digestive diseases. Where Salmonella, which is our typical food poisoning, you need to ingest as many as 1 million of these particular bacteria because they're not able to survive in a stomach acid as easily. So their ID50s are very different despite the fact that they're considered cousin bacteria. Now, the course of an infection is going to have 3 major stages, and the first stage is incubation, and this can vary considerably from just a couple days for things like the common cold and flus to years for things like HIV, Hansen's disease, which is also leprosy, and things like tuberculosis. And so this is all going to vary, and it can also vary depending on the host too. So it could be dependent on the host condition, whether they are experiencing any sort of immunocompromising situations, or the infectious dose was much larger than a typical person. And again, one of the things for leprosy and tuberculosis is that they're both mycobacterium, which grow incredibly slow. In the actual lab setting, it usually takes more than a day and up to 40 days to actually get colonies that we can actually work with, where our typical bacteria that cause disease are able to replicate every 20 to 30 minutes. So the next one is incubation period. You're typically not going to have a whole lot of signs in that person. They might start feeling a little bit of malaise, which malaise is just not feeling all that great. But it isn't until they actually get to the illness stage that they're going to experience the signs and symptoms of the disease. This is going to be where they actually are saying, okay, I'm sick, there's something wrong, I need to get it figured out. Now that other one, the malaise, is actually going to be considered what's the prodromal phase, which are just the vague symptoms. Everybody here has had that. You wake up with a small headache, you wake up with a little bit of a runny nose, you feel a little tired, whatever it might be. This is what we call the prodromal phase, right before actual disease shows itself completely as an illness. Then ultimately, as you clear the disease, or as the immune system clears the disease, we go into convalescence stage, which is just really the recuperation, recovery from the disease. You may still have a running nose, cough, whatever it might be. You may not be feeling all that great for another couple days to a couple weeks, but ultimately you're on the path to recovery. All those damage that's caused by the inflammation and whatnot is now in the stages of being repaired in this body. Now, some people are actually what we call carriers. This is going to be somebody that carries an infectious agent for their life, or for just a severe long period of time, and they have absolutely no signs and symptoms. So one of the more famous stories that we'll be talking about later is Typhoid Mary, in which that she was a carrier for Salmonella typhi, working in restaurants, and ultimately was arrested because this was before they understood that bacteria caused disease, and they didn't really know what to do with her. And so we'll be covering that story later in the semester. So the symptoms of the disease are going to show up as acute, chronic, or latent. And in this case, acute is going to be they develop quickly and last a short period of time. A cold, strep throat, flu, food poisoning. Well, all these things are what we call acute. They're going to be very fast onset, usually within a couple days of actual being infected, and then they're going to usually be cleared by the immune system pretty quickly. Other ones are going to be what we call chronic, which develop very slowly and last for months or years, and this could be things like tuberculosis, HIV, leprosy, those that we And one of the last points is that not all animals are going to work as good hosts. So one, something like Salmonella enterica, which causes food poisoning in us, actually causes systemic infection in mice. And so the virulence and the actual pathogenicity of the bacteria is vastly different in our typical gold standard mouse versus humans. Another one is something like flu. Flu does not really infect mice whatsoever, but in a ferret, it actually causes symptoms and signs very much like what we see in humans in terms of the ferret sneezing, actually, where mice don't sneeze, and so we actually get signs and symptoms that are much more similar. So sometimes we have to go off the typical gold standard mouse to actually find suitable models. Now we've taken it a step further with the recent technology advances in the last 20-30 years, where we're actually able to see the molecular side of things, and through genetic engineering from what we talked about in chapter 8, which was the plasmids found in bacteria, we can actually genetically engineer these plasmids to carry whatever genes we'd like them to carry. And so we can actually go in and take out virulence factor genes and try to actually establish disease in an animal with that gene missing. And if the bacteria suddenly loses its ability to actually cause disease, and when we actually think that we might be on to something, we put that gene back in, and if it's able to cause disease again, then we assume that that is a virulence factor. So this satisfies molecular Cox postulates in a different way than what we see with the traditional. So some characteristics of pathogens. What you're going to see as we start going through the rest of the chapters, when we get into the respiratory chapter in 21, we'll be basically following that same style the rest of the semester, is that they're going to start sounding a lot alike, and that's because there are some general characteristics of pathogens in terms of how they're actually able to establish disease. One of the gold standards, forgive me for using that word again, it's early, but essentially the toxins are a big deal. So a lot of bacteria that we're going to be talking about are able to produce toxins, which are actually what's causing damage to the host. It's not the bacteria itself, it's actually the toxins that they're producing. Others are able to colonize the mucous membranes and then produce toxins. And so you see the list here: Vibrio cholera, E. coli, which is one of the deadlier versions of E. coli, and diphtheriae. All of these are able to actually colonize the mucous membranes and cause toxins to be formed. Others are going to have camouflaging techniques, as we've seen in chapter 3 when we were going over all the different characteristics of bacteria. A lot of these are going to be carrying capsules, and these capsules prevent them from being seen by the immune system, or actually protect them against the defenses by basically blocking. And we'll see a lot of opportunities throughout the chapters of bacteria that are able to do this. And the last one is invade the host tissues, actually be able to chew through. So something like flesh-eating disease and whatnot, able to just go ahead and chew right through the tissue, and none of the innate systems really able to stop it whatsoever. Now, when we speak about the actual microbe causing infection in an individual, as I said earlier, the first thing it has to do is actually adhere to one of our cells. Otherwise, it's just going to get pushed through either the digestive tract, kicked back out of the respiratory. Somehow it's got to be able to stick. And so those molecules that are responsible for allowing the microbe to stick are called adhesions, or more specifically adhesion proteins. These are going to attach to specific receptors on the cell surface of our cells, or whatever the host may be. These adhesion molecules are going to be very specific. So as we'll see in the respiratory chapter, the microbes that cause disease in respiratory systems typically only attach to respiratory epithelium cells. A lot of times these are actually going to be located out on the tips of the pili, and that's what we call the fimbriae, basically the ends of the pili or the fimbriae, and that's where we're going to find a lot of these adhesion proteins, which makes sense because that's at the very tip. It's going to be the first thing that runs into any of the epithelial cells. And now, some of them can also be part of the capsule or other parts of cell wall proteins, but typically found on the pili. Colonization is going to be the bacteria actually growing. Once it's established an infection, it may be like with Shigella, it may only be a few of them, but as they're able to grow every 20 to 30 minutes and double, it doesn't take much more than 24 hours for a bacterial colony to be able to establish disease. And so some of the ways they can do this is growth in a biofilm. We discussed biofilms in previous chapters, but essentially this is where a bunch of bacteria are able to grow as a village almost, and they basically cover the entire colony with a mucous membrane type substance that we now call biofilms. And these are important in a lot of different microbe medical conditions, but we'll be covering them more as we go along. Cytophores are essentially gatherers of metals in our system, so a lot of the enzymes that we have, as well as the enzymes that bacteria have, they require metals to act as cofactors to make that enzyme work. And so these bacteria are going to be trying to pull magnesium and zinc and other iron, other metals out of our system for their own use. And so this is one of the ways that they can colonize. Obviously, avoiding the immune system is a big deal, and being able to compete with the normal microbiota, sometimes by producing toxins to actually kill off the microbiota, is a good strategy for some of these pathogens.